Hello, welcome to the 5th Fall 2025 Northeast Cover Crops Council webinar. I'm Victoria Ackroyd, NECCC Program Manager. My co-host is Susan Brulette, Program Manager for the Northwest Crops and Soils Program at UVM Extension. Before I introduce today's speaker, a little bit of housekeeping. If you have any questions, please feel free to throw them into the chat as they come to mind. I'll handle the question and answer moderation session at the end of the webinar. Today's session does count for one Nutrient Management CCA CEU. To get credit, please email your full name and CCA number to Susan. I'll put her email in the chat shortly, or you can just direct the information to her in the chat. This webinar is being recorded. It will be available soon. And now for today's speaker, Charlie White. Dr. White is an associate professor and extension specialist in soil fertility and nutrient management. He is broadly interested in how biogeochemical cycles, particularly those of carbon, nitrogen, and phosphorus, can be managed to promote sustainable agricultural systems. The goal of such management is to support cropping systems that are highly productive, use nutrient inputs efficiently, and have fewer nutrient losses to the environment. Recent research efforts have focused on optimizing the use of cover crop mixtures to both retain nitrogen against leaching over the winter and supply nitrogen to a subsequent cash crop. Charlie, thank you for joining us today to talk about cover crops and nutrients. You have the floor.
Great. Thank you very much, Victoria and Susan, for hosting today. And it's great to be part of this webinar series with the Cover Crop Council. So I'll go ahead and share my slide deck here, and then we'll go ahead and get started. So the topic of my presentation today is to share some work that we've been doing in Pennsylvania on developing nitrogen recommendations for corn that account for cover crops and soil organic matter. And this is a project that we've been working on for a number of years, and have, through that work, have been able to pull together a new decision support tool for growers to use. So some of the motivations for developing these new nitrogen recommendation tools is that the development of no-till farming systems and more advanced and diversified cover cropping systems has really made nitrogen management more challenging, right? So we sometimes think about cover crops as a good tool to help improve nitrogen management, and they can really do that. But we also, on the flip side, need to recognize that it might improve our management, but it also makes it more challenging. And the challenge comes in that it can be very hard to sometimes predict what the effect of the cover crop will be on nitrogen requirements for the next corn crop. Also related to the no-till management, no-till management makes things a little bit more complicated depending on what type of fertility sources we're using. We might experience more volatilization losses if we're not getting our fertilizer incorporated. No-till can also increase soil organic matter levels over time. And so what's the effect of that increase in soil organic matter in terms of a nitrogen supply, right? Does that allow us to reduce our fertilizer applications? And then sometimes you've got interactions between cover crops and no-till, right? So you might have a cover crop that ties up a lot of nitrogen initially, produces like really high biomass residue cover like this, right? Is that going to tie up nitrogen? Maybe, but then over time it's also going to build soil organic matter, and so then that's going to supply that back as some source. And so at what point does it stop immobilizing and start supplying? Like I said, it makes things more complicated. So, um, hence the need to have more advanced decision support tools that are driven by data, measurements of the soil organic matter, measurements of the cover crops, to get more site-specific recommendations. So one of the challenges with our existing or historical nitrogen recommendation methods is that they really haven't done a good job of accounting for organic matter credits or cover crop mineralization credits. So what I want to do here today is just briefly review our traditional method of generating recommendations, but then introduce a new system that we've developed here in Pennsylvania that might be of interest to other people as well. Right now, I don't really recommend it be used outside of Pennsylvania because it's calibrated for our region, but maybe it will stimulate some interest, and it's still just good to know about what we're working on. And then also I'm going to briefly touch on a recalibration of the pre-side dress soil nitrate test uh, to accommodate the use of cover crops as well. So traditional method of making nitrogen recommendations. You all are probably familiar with the old rule of thumb to fertilize with 1 pound of nitrogen per bushel of expected corn yield. But you might not be familiar with how we got that rule of thumb. So I want to just explain and share the data set. This is a data set from my predecessor, Doug Beagle, who was the nutrient management and soil fertility extension specialist prior to me. Uh, this data set represents many different experiments where they grew corn and did a response curve, so increasing doses of nitrogen, and you see where's that economic optimum response curve, and they've sorted that by what the optimum corn yield was that they were able to achieve in that trial. So from left to right, the yields are sorted from high to low. And then for each trial on the y-axis is what that economic optimum nitrogen rate was. And you can look at that and see there's a lot of variability, and there may not necessarily really be like a clear one-to-one trend where, it increases monotonically. But the way they developed that 1 pound per bushel recommendation is if you bin these into groups and say have a group, from up to 130 bushels per acre corn yield goal, there's kind of a maximum nitrogen recommendation at about 130 pounds N that is enough or more than enough for all of the observations in that group, right? All of these, by applying 130 pounds of N, would have met or exceeded the nitrogen need of these crops, right? The next group from 130 bushel to 160 bushel per acre, almost all of them, 160 pounds of N would have been enough to meet or exceed their need. And then up to 190 bushel per acre, 190 pounds, right? So we start to see now here within these categories, there becomes this one-to-one relationship. But it's a challenge because a lot of these didn't even need that much, and sometimes they needed more. And if we just have a crude recommendation like this, how do we maybe sort out the sites that need a lot less? How do we sort out the sites that might need more? And how do cover crops impact this, right? Maybe a really highly immobilizing cover crop might make it more likely that your crop needs more than this 1 pound of N. Maybe a legume crop needs a legume cover crop means it needs less than one. Maybe a long-term manure history, maybe building soil organic matter through no-till, right? So we need to get more precise moving forward. So as we started going down this road, we were looking at a lot of response curves from different experiments. Many of these are on-farm experiments. Most of these have some type of cover crop in the system as well, and we're sort of data mining these response curves, trying to understand the patterns of their behavior. And one of the things that we started to see is that 1 pound of N per bushel of corn, that 1-to-1 ratio, really doesn't seem to hold up very well, right? So here's a response trial where we only got 113 bushels of corn, and that's because it was a very dry year. It was a drought year. It took us 173 pounds of nitrogen to get that 113 bushels of corn. Here's another response trial in a different part of the state, maybe slightly drier than normal year, but still pretty good yields. We got 214 bushel corn with that same amount of nitrogen, 172 pounds of nitrogen, right? So here we used 173 to get 113 bushel. Here we used 172 to get 214 bushel, right? So it's the same amount of nitrogen needed in both of these, but very different yield goals. So that 1 pound of N really per bushel really doesn't hold up here. But what's similar between these two response curves is a property that we call the delta yield. And that's the difference between the yield at the plateau and the yield with no fertilizer added, right? Both of these curves have a 40 bushel per acre delta yield. And what we've seen through analyzing lots and lots of these response curves is that the nitrogen rate is more related to this delta yield than it is to the maximum optimum yield, right? So here's another set of response curves. Here's just to illustrate the point even more, right? Both of these have about the same optimum yield goal, right? 241 versus 250 bushels. But this response curve is after a mature wheat cover crop, right? So high biomass, high C to N ratio, lots of immobilization. And we see that that response curve gets pulled down, way down here, where we lose a lot of yield as we take fertilizer away, right? So it took 275 pounds of N to get up to 250 bushel here with this mature wheat cover crop. Here we're getting to the same yield goal, but it only took us 134 pounds of N. And that's with a young rye cover crop killed early that mineralized the nitrogen back rather than immobilize it. And so they have very different delta yields here, and the N requirement is really a reflection of this delta yield um, property of the response curve. So how do we predict delta yield? When we tried to publish this, it took us like 5 years to get this data set published because all the reviewers said, well, you can't predict delta yield. That's totally unpredictable. And so how could you develop a recommendation off of it? So to calculate delta yield, you look at the maximum attainable yield, you subtract the yield with zero and fertilizer applied, and that's your delta yield. So we have to be able to predict these two components. So this maximum attainable yield, this depends some extent on the weather, the soil type, your corn genetics, your grower management. But a grower probably knows sort of within a range of what they typically can get in a typical weather year. They've probably got lots of experience with, well fertilized crops to know what that yield goal is. So we're suggesting that you could, for planning purposes, determine that based on historical data and just take an average of your historical yield records to calculate a typical attainable yield. So the yield with zero N fertilizer applied, that depends on the soil nitrogen supply, which is coming from organic matter and your cover crop residues. And it's this microbial process of mineralization and immobilization that's occurring. And that depends on a lot of different things. Depends on soil texture, organic matter level, cover crop nitrogen content, cover crop C to N ratio, right? A lot of different things can affect that. And it is somewhat complicated, but it's not totally unpredictable. And the reason it's not unpredictable, or the reason I, don't want to use a double negative there, the reason it's predictable is because we actually know a lot about this mineralization process, and we know a lot of details, but we also can simplify it into a relatively simple model. And so what I want to present here is a fairly simple and straightforward model of mineralization, right? So on the left here, we have decomposing organic material. That might be your soil organic matter, that might be your cover crop residue, and it's got some component of carbon, it's got some component of nitrogen. Can calculate the ratio of carbon to nitrogen. That's our C to N ratio. When microbes are decomposing this material, they're after the carbon for two different reasons. One is to build their biomass, right? To multiply and divide their cells and build all of the structures they need inside their biomass. But they're also getting energy from that. And when they generate energy from that carbon, they respire that out as carbon dioxide. Now, as the microbes build their biomass, they also need to incorporate some nitrogen into their biomass to build proteins and other enzymes and other cellular structures and whatnot. So they're going to utilize some amount of that decomposing nitrogen from the residues to build their biomass. And if there's enough left over, if our C to N ratio is low enough, if we have enough nitrogen in the decomposing material relative to carbon, that's going to be released into the soil in an available form as ammonium, and then it might nitrify into nitrate if it's not taken up immediately. And that's available to feed the crop, right? And that was what we would call net mineralization. So luckily, this process, if we simplify it, it can be simplified into a relatively straightforward formula, right? We've got the amount of nitrogen mineralization is related to the amount of nitrogen in the cover crop residue multiplied by some kind of decomposition rate. And then we subtract off the efficiency, 1 minus the efficiency factor. And that efficiency factor is what proportion of the carbon goes into the microbial biomass. that tends to be dependent on soil texture and maybe some microbial community composition, uh, multiplied by the seed to N ratio of the cover crop residue, divided by the seed to N ratio of the microbes, right? And the seed to N ratio of the microbes, that's typically around 10 to 1. So, a lot of these things are measurable and somewhat predictable, constrained based on knowledge we have about this process. So we have a formula that we could use to predict that, and we can calibrate that to experimental data. So, that's the mineralization process. The backwards react process can also occur. We call that immobilization. If this cover crop residue does not have enough nitrogen in it relative to the carbon, the microbes are going to break down that carbon, build their biomass, not have enough nitrogen from the residue, so they need to scavenge it from the soil. And if that happens, if that cover crop C to N ratio gets high enough here in this formula, it turns this equation negative and you get a tie-up or some immobilization. So it's pretty, actually mathematically, it's a fairly straightforward formula to calculate this. So we've taken this formula about nitrogen mineralization and built it into a decision support tool that is calibrated to experimental data in Pennsylvania right now, but not saying that couldn't be expanded if folks wanted to do the research elsewhere. The main thing that really needs to be figured out is this decomposition rate, and that could be calculated by environment, temperature, moisture, things like that. So anyways, our decision support tool, it's calibrated for Pennsylvania, and the way it works is it first predicts the amount of yield that can be achieved with no additional fertilizer. And that is based on our mineralization model. And it takes inputs of soil, carbon, and nitrogen ratio, soil texture, cover crop nitrogen, and cover crop C to N ratio to predict that unfertilized yield. Then we have the grower determined yield based on weather, soil type, and management. That would be your historical yield goal or your maximum attainable yield in an average year. And we calculate the delta yield. And then based on a large database of response curves, we use delta yield to predict the optimum rate of nitrogen fertilizer required. So what do you need to run this tool? You need an average corn yield goal. So recommendations are designed to be made at the beginning of the growing season when the yield potential for the year is not known. So therefore, we recommend using an average yield from historical yield data to develop this input. The next thing you need are measurements of soil and cover crops. So the first input is soil texture, so sand percent and clay percent. And we have calibrated our tool to be based on a soil sample to 8-inch depth. And we recommend getting a lab measurement of soil texture. This is something you only need to do once because soil texture really doesn't change over time. That's a kind of a permanent inherent property of a soil. The next thing to measure is the total soil carbon and total soil nitrogen as a percentage. And the tool will input this total soil carbon directly, and then also you take these two values and you calculate a soil C to N ratio, and you put that into the tool. Again, we recommend this from a soil sample measured to 0 to 8-inch depth, and we really recommend a lab measurement of directly measuring the total soil carbon and nitrogen on a percentage basis. The tool needs a cover crop nitrogen content input, and that could be based on a measurement with a handheld green seeker. I'll show this in a second. And we've also are developing ways to calculate this based on drone or satellite NDVI. But maybe you could also just use sort of visual estimation as well. That could be adequate in some cases, and convert that to pounds nitrogen per acre. And you need a cover crop C to N ratio as well. And what we're currently suggesting is that instead of getting a laboratory measurement of this, people use a lookup table based on our database of C to N ratio samples. Okay, so submitting soil samples for analysis, one of the things we've learned is that there's a lot of variability in how commercial labs and university labs test soil organic matter, how they test it, how they measure it, how they report it. And because of the wide variety and also the sensitivity of our tools recommendations based on this input, we really recommend currently a standardized uh, doing it at the Penn State and Ag Analytical Lab in order to have a standardized methodology, right? I'm not saying that forever people need to do this, but currently, just sending it out to some random lab that says they're going to measure organic matter, says they're going to measure carbon, there's just too large of a variety, and labs are not transparent about how they actually do things and report things, that I'm hesitant to recommend sending it anywhere else than Penn State at this point in time. So what on our Penn State submission form, for multiple submission form for agronomic crops, we've got really handy check boxes over here under additional analyses. For total CNN, you see that costs $25, and particle size will get you your percent sand and percent clay measurements. The soil texture, that's an additional $25, right? So if you do want to send a sample elsewhere, what I would say is just verify that they're doing total carbon and total nitrogen by elemental combustion analysis. Okay, anything other than elemental combustion analysis is going to be fraught with variability and uncertainty and assumptions built in. So, like I said, you don't have to use Penn State forever, but just please use a lab that is measuring total carbon and nitrogen with elemental combustion analysis. Okay, so cover crop nitrogen measurements. One of the tools that we've developed over time, well, we didn't develop the tool, but we developed this application of the tool, is to use a handheld green seeker sensor to walk over a cover crop canopy. So here we've got someone walking the sensor over a cereal rye cover crop, and they're getting this NDVI value here. And so we've done this over hundreds of different cover crop types and developed a calibration. Go to this link here. We have a fact sheet about it to convert that NDVI number into pounds nitrogen per acre. We're also working on releasing a tool that would use satellite NDVI or drone NDVI, or also NDRE is the normalized difference red edge. And you could use any of those metrics, and we'll have a tool that will predict the cover crop nitrogen from those. So that's kind of in prototype development now, but I hope in the next year we'd be able to release that as well. The cover crop C to N ratio, I mentioned using a lookup table based on historical database. This is something that Kathleen Arrington pulled together. I saw she was in the attendee list here, so thanks, Kathleen, for pulling this together from a very large data set of many, many years of cover crop. So we have the average C to N ratio, and then the typical range of values based on species and mixtures and their termination timing at maturity at termination time. And we've also observed some things based on like whether manure was applied to the cover crops or not, or whether the field has a long-term history of manure applications, right? That tends to increases nitrogen available to the cover crop and lowers the C to N ratio. So we have those kind of adjustment factors suggested as well based on the manure history. So this lookup table is part of a fact sheet that we've developed that describes our whole tool. And if you want more information about that, highly recommend going to this fact sheet, and you'll see all the background of everything that I've explained today is in that fact sheet, including this lookup table. Okay, so here's the tool. The tool can be accessed through this link here, and there's a couple different formats. There's a web-based application that I'll show you in a second that has a nice graphical interface. There's also a spreadsheet format, so if you wanted to do this for many different fields and just sort of copy and paste from a spreadsheet, carbon and nitrogen from lots of different fields. Or another application of this spreadsheet would be to do variable rate management. So let's say you have hundreds of different polygons for a field that represents different cover crop nitrogen and C to N ratios, like maybe you had a satellite image or drone image and you wanted to do sort of pixel by pixel recommendations. This spreadsheet tool is a way that we've done that as well. And then you get a nitrogen recommendation for each zone of the field, and you can upload that into a prescription file for a variable rate application, something like that. So we've got both of these options available here. Okay, here's the recommendation tool. And let me just pause the video real quick here if I can. Yeah, there we go. Oh, no, that's not paused. All right. I think this is paused here. Okay. So let me just explain what's going on here. So on the left here, we have the inputs, right? The things I've talked about: corn yield goal, clay, sand content, soil carbon, that calculated soil C to N ratio. Here's the cover crop information for winter killed cover crop nitrogen, spring cover crop nitrogen, spring cover crop C to N ratio. Those are the inputs. Then over on the right-hand side, what the tool is actually predicting is it's predicting the unfertilized corn yield, right, based on that mineralization model that we've calibrated for Pennsylvania. That mineralization model is driven by all of these inputs over here on the left. And then we've got your corn yield goal, right? That sets your kind of your yield plateau. And then in between those points, right, that's our delta yield, and that predicts the optimum agronomic optimum rate of nitrogen. And then the last thing that's built into this tool is also economics, right? So once we know these two endpoints of the curve, we can draw a quadratic line, and then that allows us to look at economic rates of nitrogen, right? So actually the tool below the screen here has price of corn and price of nitrogen fertilizer that you can input, and that's going to change where the actual economic rate of nitrogen is, right? If your fertilizer is very expensive, it's going to pull that economic optimum nitrogen rate down depending on the response curve. So, what the response curve predicts ultimately is the additional amount of recommended nitrogen needed to reach your economic yield goal. And that additional recommended nitrogen, you need to then take that and put your agronomist hat on and say, how are we going to get that nitrogen, right? That could be from manure applications in the spring of the year. It could be from some starter fertilizer. It could be from some side dress fertilizer, right? So you need to think agronomically about what combination of different nutrient sources are you going to put together in some type of a program to ultimately meet those needs. And I'll give you some examples of how we've, can look at that through some on-farm response trials that we've done. Okay, so let's watch this as we adjust the inputs here. Our delta yield is changing, our response curve is changing, right? As we change the soil texture content, that's actually changing the microbial carbon use efficiency, and that changes up here what we call the critical C to N ratio. So if your cover crop gets above that C to N ratio, it'll start immobilizing nitrogen. The other thing we can see here is what's called the cover crop yield credit, and that is how much the cover crop is changing this unfertilized yield value, right? If you have a positive cover crop credit, it's bringing that unfertilized yield value up. If you have immobilization occurring, it's actually bringing that unfertilized yield down. And so as we increase that C to N ratio here, right, it can go, the cover crop yield credit can go negative, right? And so it's actually pulling that unfertilized yield down. And any adjustments of this unfertilized yield will ultimately translate into changes in your additional nitrogen recommendation. Okay, I also want to take a moment to give a shout out to another adaptive test that is the pre-side dress soil nitrate test. And this would be done in season at the time when the corn is growing. We recommend about when the corn is 12 inches tall that you would pull this soil sample. And historically, there have been formulas for how to calculate the amount of additional side dress nitrogen needed based on this PSNT result. Historically, if the PSNT level was over 21 part per million, no additional side dress N was recommended. If it was less than 21 part per million, then at least in Pennsylvania, we had a formula that was calibrated. And my predecessors put together this formula. But one of the things we saw, and this was done in the late 1990s, one of the things we see with modern production is that it doesn't have cover crops in this, right, as the different factors and whatnot. So we wanted to see, okay, should we recalibrate this to include cover crop systems and no-till systems? So we did that, and we found that we needed a slightly different calibration. So we've got a new formula here. You take your corn yield goal in bushels per acre multiplied by 0.72, and then you subtract off the product of this coefficient 5.1 times your PSNT value in nitrate. So if your PSNT level is really high, multiply by 5.1 and subtract it off, at some point, this formula goes negative. At the point that this goes negative, that means you've got sufficient nitrogen mineralizing from soil organic matter and cover crop residues and manure that you don't need to side dress any nitrogen. But if you get a positive value from this formula, then you need to side dress that amount of nitrogen. The other thing we saw with our data set is that there was kind of a fudge factor, I'll call it, if the cover crop was a mixture of plant families. So we had to add 50 pounds of nitrogen on to whatever was calculated up here when the cover crop was a mixture. And we think what's happening with this is that in mixed cover crop stands, the legume component tends to mineralize very quickly early in the season, and that mineralization leads to a higher PSNT result at the time of sampling. Meanwhile, the grass component of the mixtures is taking longer to break down, and as it's breaking down, it's immobilizing nitrogen. So you got a little bit of a late season immobilization effect going on that isn't measured at the time of this PSNT sampling. And so we think that's the reason why our data set suggested that an additional 50 pounds of nitrogen needed to be added on. But again, I want to reiterate that's our speculation, but this coefficient here is all data driven based on the modeling that we did. That number popped up for mixtures. So we're just kind of speculating why that might be. Okay, so I want to conclude here with just some examples from some on-farm experimentation that we've done to verify and validate that these tools are working well. And these data are all from a PhD graduate student in my group, Andrew Lefevre. So I'm very appreciative of his efforts to run these trials and collect the data that we're sharing here today. So I'm just going to share one example of the details of what went in. So this was a response trial from Manheim, Pennsylvania, that's in Lancaster County. We've got our inputs, right? Our corn yield goal of 225 bushels, soil texture, 1.5% soil carbon to nitrogen ratio of 9 to 1, cover crop N content of 33. This was a cereal rye cover crop, killed fairly young, so it didn't accumulate a lot of nitrogen, but its C to N ratio was also relatively low, and that means it's going to be a mineralizing cover crop. And then corn price and nitrogen cost in the year of the trial, which was 2023. So some other details from this site. After cover crop termination, liquid swine manure was injected at 5,000 gallons per acre. So we're going to have to take some kind of credit of that nitrogen availability. That's going to go towards meeting our additional recommended nitrogen that the tool suggests. So we use agronomy guide calculations based on availability factors. So for the ammonium N content, it was 12 pounds N per 1,000 gallons, 80% available because it was injected. That's 48 pounds of N available. The organic N component, 0.5 available, 26 pounds per 1,000 gallons. That's 65 pounds of the organic component that's available. So our total spring manure credit is 113 pounds per acre. And that's going to go towards meeting our additional recommended amount. So when we plug all this data into our tool, we get this response curve here. It's suggesting our economic optimum nitrogen rate is 146 pounds of N that needs to be supplied. We need to account for this spring manure credit of 113 pounds. We subtract that off, and that gives us an additional balance that we need to apply with fertilizer. So here's a table that kind of goes through doing the calculation of those balances. Additional recommended nitrogen of 146 pounds of N. We subtract off the spring manure credit. The farmer also wanted to plant with a little bit of starter, so 35 pounds of N was used as a starter, and then the net amount of recommended N after the manure and the starter, we're already negative here. We're negative by 2 pounds per acre. So we would say the amount of additional side dressing that would be needed, because agronomically the farmer's like, okay, I want to split my nitrogen up between some manure nitrogen, some starter nitrogen, and some side dress nitrogen. The grower's standard side dress nitrogen is 85 pounds N per acre, but based on this balance, the end tool is saying we don't need to actually side dress any additional. We've met the need with just the manure and the starter. When we collected a PSNT test, we got 56 part per million nitrate, and based on the yield goal and that formula, it came out to not need any side dress either. So, the grower's doing his standard thing, and we've got these tools saying you don't need to do that. Well, what do you think the response curve looked like? So this is the response curve, right? We side dress different rates of nitrogen on that corn, and we see there's no response, and the farmer has met their yield goal of, they reached 229 bushel corn without any side dressing, and that's a flat line there. So we've done these types of validation trials on a number of different sites over the years in Lancaster County, and we're doing them elsewhere as well, but this is just data set from Lancaster County. You can see the range of previous crops, the range of cover crop types, the range of cover crop management. I mean, we've kind of got it all, right? We've got early burn downs, late burn downs, planted green, rilage harvest. We've got a variety of different recent manure application types, right? It's pretty representative of the diversity of manure and cover crop management practices that we have here in Pennsylvania and probably in the broader Mid-Atlantic region and Northeast. So here's the 7 response trials, the grower's yield goal, what they actually achieved at the yield plateau. And then we have the nitrogen tool, that's the online interface I just shared, and then the PSNT recommendation. And then we have the economic optimum side dress nitrogen recommendation. And anywhere you see a green shaded cell, we felt like that either N tool or PSNT was an accurate close to accurate within, small margins of error uh, recommendation relative to what the the experimental results were. So we see there's a lot of green cells, and the red cells, what happened there, right? So this first red cell with the PSNT, this farmer planted corn late, and so we collected a PSNT relatively late compared to our other trials. We're wondering if that timing of the PSNT sample might have led to a high nitrate level and a low recommended amount of nitrogen compared to what the grower actually needed based on the experimental results. These red cells here was a farm where there was a pretty bad drought during the growing season, and the optimum yield goal, or the yield that was achieved, did not reach their yield goal due to the dry weather. So they only got 164 bushels. Typically, they're getting on average 230 bushels. And so we developed these recommendations with the N tool and the PSNT based on this 230 bushel crop. It's calling for 70 pounds of side dress with the N tool, 63 pounds of side dress with the PSNT. But because of the suppressed yields, there was no response to the additional side dress nitrogen. So, there's a couple outliers here that weren't accurately predicted, but we, maybe have some indications of what went wrong at those sites. But lots of other sites where it was well predicted. And if we exclude the site that was a dry year, and we use, say, the greater of either the N tool or the PSNT recommendation, and we compare that to the grower's standard side dress rate, on average, what we're seeing is that on average, the farmers could reduce 48 pounds of nitrogen. And based on a recent fertilizer price, could save about $43 per acre on average by using these tools. So it just speaks to the opportunity for reducing nitrogen recommendations, especially in fields that have a heavy manure history and cover cropping. Over lots of different sites, we've seen a pretty strong relationship between our predicted economic optimum nitrogen rate based on the nitrogen tool and the observed economic optimum nitrogen rate. So there's a very close to one-to-one relationship here, and pretty high R squared, and scatter around that line is sort of what we might expect, typical level of accuracy, maybe accurate to be within about, say, maybe 30 pounds of nitrogen or so. Most of these are plus or minus that. So we think we're having pretty good results with this tool so far. So to conclude here, and then we can take some questions, right? We need an adaptive approach. There's not a one size fits all. That 1 pound nitrogen per bushel is probably a coefficient that would be good to do away with, but sometimes it's hard to shake off history. Recommendations can be improved with these new tools and updated calibrations and interpretations. Using these tools in combination will probably be a good strategy, right? So use the nitrogen tool at the beginning of the season to help plan your strategy. Maybe check in mid-season with a PSNT test. There's not enough time today to talk about the cornstalk nitrate test, but that's a great end of season test to see whether your nitrogen rate was too low, optimum, or too high. Um, and then all of this talk so far has been about the right rate, but still remember those other four R's, right? The right source, the right time, the right place, right? All of these things need to be fine-tuned and optimized so that you get the most efficient utilization of the nutrients that you do apply. So be glad to take questions now. We've got some additional resources. Also just want to recognize a really large group of people that over many years has contributed towards putting together this work. Lots of collaborating farmers, many students, postdocs, and technicians, lots of other faculty uh, here at Penn State have been involved in this, and lots of funding agencies supporting the work as well. So this is just kind of a visual to show the type of resources that our farmer collaborators and industry collaborators mobilize to get these sites done, right? This is a common view at harvest from one of our on-farm experiments. So I'll stop here and see if there's any questions.
Thank you so much, Charlie. Um, I mean, you guys are welcome to put questions in the chat, or you can raise your hand, or you can just unmute yourself. Any questions? I have a question. I
Yeah, thanks,
actually
Victoria.
work a lot with decision support tools myself, though I tend to do more with what we call expert opinion-based type tools rather than the empirical type tools that you're clearly talking about here, that are based on field work that's quantitative rather than qualitative. Are you aware of some of the other nitrogen cover crop tools that are out there? Can you comment on similarities, differences?
Yeah, definitely. So, well, I know the Northeast Cover Crop Council has been working very hard on nitrogen credits from cover crop residues, and you might need to correct me if I misstate anything here, Victoria, because I think you know that tool pretty well. But my understanding is that tool you submit a cover crop sample to a lab, right, and you have a forage analysis done. And then based on the quality characteristics of that cover crop, including things like protein, but then also cellulose and hemicellulose and degradability and things like that, there's a decision support tool that predicts the nitrogen credit or the nitrogen mineralization from that cover crop residue. And I think one of the real advantages to that tool is that you get like more refined information on the release patterns of the cover crop, the nitrogen release patterns. I think it's also taking into account more precise weather so you can use it over a broader range, right? I think it's calibrated all up and down the East
East
Coast,
Coast.
if
Yep.
not further than that, right? So you can dial into a weather station and it'll, very accurately predict the decomposition rate. So, I've said a few times our tool is only calibrated for Pennsylvania. That's a real disadvantage, but I think the one that the Cover Crop Council has is broader. I guess one difference between this tool that I've described and the Cover Crop Council tool, I'm not sure whether the Cover Crop Council one takes into account soil organic matter pools. I don't
Mm.
know. Does it account for that?
Off the top of my head, I unfortunately couldn't tell you. I'm not really involved with the guts on that tool. But I know that SOM is certainly key to mineralizable nitrogen. I'd be amazed if it doesn't take that into account.
Yeah. So I think that's another just part of the component. And what we've seen from calibration of our tool is that the biggest driver of the nitrogen recommendation is not actually the cover crop residue. It's more I mean, that will change it a little bit, but what really can make a dramatic effect is the soil carbon and the soil C to N ratio. So I think, and it's, as you're evaluating different decision support tools, it's really critical to think about what that soil organic matter pool is, because I think relative to the cover crop, it's a larger contributor to the nitrogen supply. Still, it's nice to know that whether your cover crop residue is going to tie up nitrogen or mineralize nitrogen and how much, that's still valuable. But knowing also the uh, the soil organic matter component as well. So one other tool that I'll just mention in comparison is a model like the AdaptN model, right? So AdaptN is a computer model,
Mhm.
and the real advantage of AdaptN, in my mind, is it takes into account the actual growing season weather patterns, and it takes into account potential losses through leaching, denitrification, or volatilization. And so the model I shared for Pennsylvania, that's based on average growing season, and it doesn't, other than maybe accounting for different manure availability factors with our agronomy guide tables, it doesn't really detect whether fertilizer you put out at planting leached or denitrified. It doesn't calculate whether urea broadcast on the soil surface without a urease inhibitor volatilized based on the weather that happened in those last 2 year or 2 weeks, right? So something like AdaptN would take into that site-specific, year-specific weather patterns and then help fine-tune your
Oh, that is a
yeah,
long-term wish of mine to be able to
yeah,
take into account real-time weather patterns.
yeah, yeah,
So I did go to our about page really quickly. I'm just naturally a little bit shy about speaking off the top of my head. We do pull soil organic matter in CCN Calc from web soil survey database. So obviously, if a field has been super intensively managed, the organic matter number could differ from what Sergo thinks it is.
yeah, yeah. Okay.
Will say you did mention, and you're absolutely correct, the need to send samples to a lab is just ugh. And so, like you, we are working to make it so that we can use satellite imagery to pull nitrogen estimation and get rid of that need for the destructive biomass harvesting, or I mean, non-destructive if you're using like I think you said it's a green seeker, the handheld NDVI. Um,
Yeah.
I think that's a common goal. I wonder if there's any actual tools that currently are out that do that actively instead of people working to get them there. But
Yeah, yeah. So, and I see Ayana has a question
yes,
in here too
yes.
about availability of that satellite imagery and using that in ArcGIS. So one tool that's publicly available now and is free, at least the last I checked, is a tool called OneSoil. I think
Hmm.
you go to onesoil.ai and you can you create an account, you map your fields, and then within that field boundary, they pull up satellite imagery. I think it's publicly available satellite imagery, and you can get a field average NDVI value, or you can sort of hover your mouse around inside the field and see what each pixel is to, get a sense of the variability. So that's a way to get NDVI data for a field average. You can't really export maps from that software, so that would be, a limitation. But you could certainly get a field average NDVI from OneSoil. And then I think there are some more advanced agronomy software packages that do provide uh, access to satellite imagery that you could create maps from, like maybe Ag Leader's SMS software, or I think the company that runs the AdaptN model is EverAg. I think they have satellite imagery that you could pull in, but I'm not super familiar with those tools. But I think if you sort of talk to some of the maybe leading edge crop consultants that use some of those digital tools, they probably would be able to access those through some other services they subscribe to.
Thank you. Any other questions? I think I just saw the chat move, so let's see. Ah, thank you. You're welcome. I'm sure Charlie says you're welcome too, Ayana.
Yes, definitely.
Well, not so much related to nitrogen, but just research in general. One of my favorite questions to ask is always, you know, has anything unexpected happened in the field or come out of this research? Just, for example, the folks that are doing hairy vetch breeding discovered that they've got a cryptic species that's along for the ride in with the hairy vetch germplasm, and they fortuitously discovered that accidentally. Anything of that nature that you could comment on, or have you been lucky enough for your research to go smoothly all the time?
Yeah, yeah. Well, no, nothing is ever completely smooth. There's always bumps in the road and learning opportunities. I would say, I mean, one thing that overall surprised me is that once we put together the model from these discrete parts, right? So the way the model works with fine-tuning all of those inputs, those are all sort of discrete submodels that we've calibrated from more isolated data sets. Um, when we sort of put it all together, one thing that really did surprise me was, I guess, the really mi relatively minor effect that the cover crops had on the final optimum nitrogen recommendation. And I mentioned this earlier that it really seemed to be like the soil organic matter was the big driver. And once you account for the soil organic matter, the cover crop is just sort of like a pretty minor adjustment. Um, now, within a range. Now, obviously, you can grow a huge cereal wide cover crop with a seed end ratio of 40, and like that is going to have a big impact. But um, a lot of times if that cover crop, if its C to N ratio is close to that, what I said was that critical C to N ratio around 20 to 1, and a
Hmm.
lot of cover crops do end up pretty close to that 20 to 1, they really have a very neutral effect, right? If you're at right at that critical C to N ratio, it's neither going to mineralize nitrogen nor immobilize nitrogen, and it's neither going to supply nor tie up, and it's basically a neutral effect. And so I think the more often than not, the cover crop effect is neutral to very minimal in terms of the overall nitrogen recommendation. And I think that that kind of surprised me because going into it, I thought we're doing this because cover crops are having a huge effect on the nitrogen requirements. And it's like,
Yep.
oh, wow, at the end of the day, they're actually kind of minor most of the time.
Very cool. Thank you. And yeah, that is surprising and maybe a little bit unfortunate. It makes me wonder if we should be managing the cover crops differently or if we're actually, thankful that it's not completely skewing the system so long as it's within a certain set of parameters.
Yeah, it's hard to know how to think of that, but also like I think it speaks to the importance of long-term cover cropping, right? So
Mhm.
if your cover cropping is over time building soil organic matter, and if it's recycling nutrients, right, you're building soil organic matter over time. And so I think the big impact of the cover crops might not be like year to year
Mhm.
changes, but like
Yep.
long-term building soil organic matter pools
To support
that
the
are gonna
nutrient.
ultimately mineralize out through that organic matter pool. That's probably the biggest impact that cover crops can have long term.
Very cool. Any other questions? Okay, well, with that said, then I'll say thank you again, Charlie, for speaking with us today. Um, it's been a great talk. I hope everybody has gotten a lot out of it. We do hope to see you next week to hear from Daniela Carrijo at Penn State and Kara Peterson at University of Maryland. They will be talking about cover crops and water dynamics. So have a great afternoon, everybody. Enjoy your lunch, and I'm gonna stop recording.